So, welcome to the seminar of the Southern California Environmental Health Sciences Center. We have a great talk today, and I'll just mention that there'll be an opportunity in the courtyard to talk to Cathrine afterwards. I'm not going to introduce her because Jesse and Carrie are hosting her, and Jesse's going to do that. For any of you that I don't know, I'm Rob McConnell. I'm the director of the center, and it's great to have you here.
Yeah, so it's my pleasure today to introduce Dr. Cathrine Hoyo. Dr. Hoyo is the Goodnight Innovation Distinguished Chair and Professor in the Department of Biological Sciences at North Carolina State University. She's the co-director of the Integrated Health Sciences Facility Core for the NIEHS P30 Environmental Health Sciences Center at NC State. And she directs the Epigenetics Cancer in the Environment Laboratory. She obtained her bachelor's in University of Sierra Leone before getting her master's in epidemiology and biostatistics at UC Berkeley and PhD in epidemiology from the University of North Carolina. And her research now focuses on environmental health disparities research, global health research, and the developmental origins of obesity and cancer. She leads several large-scale cohort studies, including the Southern Liver Health Study, or STRIVE, which is one of the NCI and NIEHS-funded UG3 cohorts for environmental exposures and cancer risk. And she also leads the Newborn Epigenetics Study, or NEST, which is a birth cohort study in North Carolina focused on exploring how early life environmental exposures and nutrition impact DNA methylation profiles. And I think that's going to be mostly what her talk's about today. So without further ado,
yep.
You take the word.
Thank you. Thank you so much, Jesse. The reason I accepted the invitation was because I'm really interested in collaboration. You know, there are two types of epidemiologists, those that collect a lot of data and seek collaborations, and those that go out and seek collaborations with data that are already collected. So we do have reasonable amounts of data and specimens in order to understand, to use that to understand how the environment impacts health outcomes throughout the life course. So I wanted to have that in mind as I go through the talk, specifically because I really am seeking collaborations right now. I do have one budding one with Jesse, hopefully, and I have been talking to Carrie about maybe pursuing others, and there's room for me. So I'm going to start first by talking about who the people are that actually made this possible. So I structured this so that there is a research manager who oversees the logistics, because these are two really large projects, and she oversees the study managers and the field staff. And then there is the molecular geneticist who is in charge of all the molecular work that we are doing, that I'm going to talk some about. And the person that Jireja Jima is actually an interesting one, because he wasn't a PhD student when he came in. But when we started our P30 center, one thing we realized was that we would need a lot of bioinformatics support, and we needed a full-time position. Right? And so he was hired. Actually, he came with us from Duke in order to fill that position. And when he got there, he's been so successful with the collaborations. Now he decided he wanted to do a PhD himself. So, and then with the bioinformatics work that I'm going to talk about, we also have had to hire an additional informaticist. who just finished a PhD at UVA, and really he is developing pipelines for this big data that we are collecting. And then we have Ali, who is a neurobiologist by training, but really she's responsible for all the specimen husbandry that given the line projects that we can. And then Adriana, who is working remotely, and it helps students with their papers on a day-to-day basis. So these are the students, the PhD students that are in different disciplines, as you can see, and our funding really comes from NIH and other institutes. And it does include that particularly because this would not have been, the work I'm going to talk about would not have been possible without that. And then we also have additional collaborators in the developmental origins of adult disease space. And these include the statisticians who are both at NC State, and it includes molecular geneticists and people who study social stressors, as well as those that are studying specific diseases. For example, we have a hepatologist who is helping with the liver studies, and then we have a neurologist who's helping with some of the work we've just started on Alzheimer's disease, and I'm going to share that with you. And it's all really in the context of this imprint control agents. And then we also are beginning to work with an anesthesiologist who is really interested in seeing how these epigenetic marks can be shifted by the massive administration of anesthesia that they have to do during the delivery process. And our center also has a relationship with the Research Triangle Institute because we did require an environmental chemist who really is focused on metabolomics, and that's James Harrington. So my talk is going to be about really, I wanted to first provide you a brief synopsis of the one of the studies that Jesse talked about, which is the newborn epigenetic study. This started maybe around 2006, 2007, when I was still at Duke University. And it has continued to this day as we follow children. Again, what made this possible are multifaceted collaborations that will find some aspects, maybe not all of them, but we've been able to follow those children for quite some time. And then talk about the challenges that we actually see in this space as we think about what kind of policy could even help our community partners advocate for, right? And this is what led us to thinking about these DNA methylation marks that probably could help us solve some of these challenges that I'm going to talk about. And then I'm going to end with this Illumina array that has just come out that is going to help us maybe solve some of those problems if you do agree with my thesis in the first place. So the idea here is that the reason we started this newborn epigenetic study was the idea that early exposures and obesity are mediated by some molecular responses. Some of them we can actually capture if we know where to look. That they do scalp the epigenome, and some of those marks are erased and you never see them again, but there are some that we can find, especially in these periods that are early enough that a questionnaire cannot possibly reach. And that results in resilience or disease risk, and that this would happen during the life course at some point. So this is the summary of the study that I'm talking about. We now maybe have, out of the 2,500 children that we enrolled somewhere between 2006 and 2011, we now have about 1,000 or 1,200 children left in the cohort. What I wanted to highlight was the fact that the children are actually multi-ethnic. We made sure that we had as few exclusion criteria as possible because we also wanted to estimate some exposure, some exposure prevalences in this population. So the median age at enrollment was about maybe 10 to 12 weeks gestation. One of the things we really wanted to understand was environmental contaminants. What environmental contaminants were out there in the community we lived in? This is a map of Durham County here. This is a map of Durham County. See here, in the first side of this is looking at the street addresses after we measured those contaminants, which were lead, cadmium, and arsenic and mercury. We then put them on a map to look at, and the reason we did that was because there were some people that looked like they had really, really high levels. And so we're trying to figure out, who are these people? So then, this is back in 2016, we then put those onto the street addresses, but then because the street addresses are not really evenly distributed in space, we decided to do that cluster analysis that says, what is the probability that this is clustered, right? Now, this places are behind the courthouse. Nobody lives, right? So what we are seeing here is the probability that there's a cluster, right? And what we're saying, and that is the heat map that says the probability is 99%, and the probability decreases with lower intensity. And from this, what we're seeing is that we have lead exposure with 3 clusters where we have very high confidence that we have clusters. And then we have cadmium. And then when we look at these two, what we are seeing is one of those clusters actually does overlap, right? And then if you zoom in on those 3 lead clusters, what we are seeing is what we were seeing before, that you have a cadmium cluster that is on top of the lead cluster. And when you take the census tracts, these are the census tracts where that community is. And what you're seeing is that it's not very much like the population we saw, which was almost 50% African American, 50% white, and a few Hispanics. In fact, we have this kind of reversed. And those are the people who live in those communities. So now, when you start looking at where is this coming from, right? The first thing is, where is this coming from? So we go back. Now we have soil chemists. And they decide, you know what, let's get some house tests and let's get some handworks, but we also want some soil from the garden where they pump their cars. And then we correlate that with the urine and blood that we have. And as you can see, those levels are really, really high, right? Even when you are looking at those blood samples, these are really, really high levels. We know about lead from the work of Irva Hertzfischer, who was at UNC at the time, and she found higher lead levels in these Durham communities. What we didn't know was cadmium. So then there's separate studies that showing different outcomes with each one of these contaminants. So what we are seeing here is that, yes, these are associated with maternal exposure, elevated levels are associated with lower birth weight, we expected. But what we didn't expect was obesity. We did not know that it would be, that cadmium would be associated with obesity. We did not know that it would be associated with liver fat in children. And this liver fat, we actually measured using FibroScan in a subpopulation. And these data are already covered. The biggest problem we have here is structural confounding, right? You just saw where these people are coming from. Entering a variable called socioeconomic status is not going to help us, right? So what do we do then? I went to my neighbors, right? I'm in the basic science department. I went to my neighbors who are toxicologists and said, you know, I really want to publish this paper, but I want to make sure that these relationships make sense. Anyway, so they started these experiments where they took the interquartile range of our exposed population and said, okay, this is what we're going to dose our animals in. So if you look at this, these are this we expose it 6 hours post fertilization to account for to mimic the human, right? So now you have the controls left and right side, and then the blood exposure right and left side, and then you have the cadmium, the right and left side, and what is apparent actually significantly is that they did an oil staining, and what we are seeing is significantly much higher level of fat in this population. So we were able to publish this paper with the human as well as this data, so that we would be able to at least have some confidence that we were a little bit beyond confounding. I mean, the problems remain, right? So then what we decided to do at that point was, okay, why don't we look at the literature, right? Where are we? And what we found was that in vivo and in vitro models, the data are very consistent. Cadmium is an obesogen. Cadmium does contribute to these problems, right? If you let animals live long enough, right? But what in humans, what we found of the 36 studies that we looked at that published in the last 20 years, now only 50% could agree. So taking this type of information to your community makes it very difficult for them to do anything about it. So we took this information, the information I showed you before, this information, together with the human information I just showed you. And the mayor was sitting there together with the state legislature, legislative person, and they said, you're telling me that your human studies don't agree, but your animal studies do agree, and you want us to spend money based on animal models, right? So then it made me realize, we are not, I think what we need is something that, because as you think about exposures, and then you think about diseases, there is an intermediate endpoint that you really want to look at. And if that is consistent, because outcomes are going to vary and people are going to be differentially resilient, right? So you end up with, depending on the population you have, you may not find any relationships. So that was the biggest problem we had. Our studies do not agree. Okay, the legislature does not want to do anything about this for the Durham community, right? So, but then we also do have other problems, right? Because we tend to measure these exposures once, but the effects of stressors also depend on a lot of other things. From the studies of the Dutch famine, we learned that it depends on the window of exposure. We knew that those that were exposed in the first trimester ended up with metabolic problems, and those that were exposed in the third trimester ended up with schizophrenia, right? We know that. So, and yet we can only measure once because that's all we can afford, right? And then there is the exposure of past generations that we are unable to account for. This Avon study and the Swedish study have shown that if you have an exposure, some of it is actually experienced by future generations. And then we also know that the dose sometimes may or may not matter. This study was conducted, the study that was conducted in the Gambia by Rob Waterland at Baylor was showing that, in fact, when you have like a very small difference between the season of conception, where people are either harvesting crop or people are harvesting and eating a lot more, or they are planting and using a lot more energy and therefore less, you actually have large differences that contribute to preterm birth that are quite significant. So we know that we have these problems. The other problems that we do have are the fact that these are historical exposures, and sometimes we're trying to measure them and look at them in relation to outcomes, right? They're historical. We don't know how long people have had them for, and we can only measure single classes at a time. Like an ICP-MS is going to measure metals. It's not going to measure PFAS. Now you need another instrument. You need some more money to do it. So, and also the fact that these chemicals, they do often co-occur. And sometimes, as you have seen in the data with in Durham County, sometimes they co-occur with social stresses that we haven't even measured. And we are not able to actually account for that. And then the fact that whether it is internal or external dose that we're using, we really do not understand these geographic differences and the ethnic contributions to these factors. So the question then becomes, what if we identify patterns of environmentally responsive, differentially methylated regions, right? Are such patterns useful to infer past exposure? So you are talking about that myth, this, where are the differentially methylated patterns? Because what we do know about DNA methylation and other epigenetic marks is that they change necessarily in response to exposure. We want them to, right? But so how do you then use them as archives of exposure? So the idea here is that you have these chemical stressors that could be chemical or non-chemical, and that there will be epigenetic programming that occurs because we are sensing a stressor. And that stressor is going to nudge the epigenome in a very specific way to record that response, and that it is those cumulative effects that then cause the adaptation that has been linked to these outcomes. So I know that there are arrays out there that people are using, and a lot of them tend to be cover 3 to 5% of the genome. And it was very difficult for us to think about using something like that and then repeat. So they're cheaper, but not that much cheaper. So we decided to go whole genome. And this is my neighbor, again, the collaboration. And this is Dureje, the Peter bioinformatics person. And this is the guy in my lab. So what we did was take those maternal people who were highly exposed to cadmium and those that were not in the mother, and then look at DNA methylation differences in both the mothers and the youngsters. Right? And what we found was that nearly 2,000 differentially methylated regions that were different enough by the 10-15% for us to include. And in children, we still had some. And the overlap was nearly 100, suggesting that, well, this is just measured once, but then you are assuming that what you see in children is probably a subset of what you see in the mothers, and those 98 suggest that. So the problem with this type of data is that you do not know what the cause and effect is. Right? So DNA methylation marks, we know they vary by cell type. We know that. In fact, if we want to know the biggest predictor of DNA methylation, it is the cell type. And also the temporal ambiguity that I have been talking about, which is you have a case control study, or cross-sectional study, and you do not know what caused what, because these methylation marks will respond to the environment, right? And the fact that DNA methylation marks add and remove methyl groups, right? They do that frequently. The data on how stable that process is over time are very, very limited. In fact, they're limited to like maybe 20, 30 people, where investigators are really trying to do this on the side, not really funded themselves. So this is why we arrived at these marks. These are not the only regions of the epigenome that are stable. There are also, I know Rob Wetterland at Baylor is working on metastable epialleles, which are another set, but we selected this because they have been previously linked to disease. So these methylation marks are found all over the chromosome, actually they're all over the genome. And they are critical for appropriate expression of genes. In fact, these genes are monoallelically expressed, and what controls that monoallelic expression is the DNA methylation. And that DNA methylation is established very, very early in life so that it is similar across tissues. By the time cell differentiation occurs, these methylation marks are already set. And these methylation marks are also, they are appropriate for, we have thought of them as appropriate for chemical and non-chemical structures, as long as they are those marks that are established early. Because we also do know that when methylation is lost in those regions, you lose genomic imprinting. You lose that monoallelic expression. You end up producing a gene which is supposed to be, whose dose is controlled by DNA methylation. You start producing it from different alleles. And what we also do know because of that is that we do have a point where we expect DNA methylation to be. You actually do expect it to be 100% in one parental allele and 0%, give or take, in another parental allele. So you expect them to be about 50%, right? And that value is actually beyond anything we can think about that we do now. We do not have that with metastability. This is the first imprinted gene that was ever. Whether this is a cartoon for it, this is the idea of proof. In fact, when I first looked at this literature, it had been published by Andrew Feinberg from Hopkins, showing its relationship with that when you lose DNA methylation, you actually it contributes to colon cancer. But what I'm showing you here is that you usually have one allele. So this is controlled from two regions that we know about. One is the IGF-2 and upstream of that is the H19. One of them is paternally expressed. The H19 is a tumor suppressor and it's maternally. And you're expecting that when there is methylation, or when methylation is lost, you would start having biallelic expression in this. And we have seen from the Dutch family, for example, this is the region they looked at. The first, actually, they looked at that region. And what they were able to show 60 years later, 60 years after the Dutch family, they were able to go back to the people and find that these were actually stable. And what they're showing here is that they are different. There is hypomethylation in this region when you compare them to the same sex. As I said, it's been reported in adult cancer and in relation, and it's also been shown in the caloric famine study, which is these guys. We also showed that it was related to obesity at age 20 months using the next cohort, but the children are much younger. And we also demonstrated the associated with cigarette smoking. So that region also, the relationship is not specific. It seems like they're also related to whether it's antidepressant medication or depression itself. Another region that looks exactly, almost exactly actually like the one I just showed you, it controls this kind of bunch of a cluster of imprinted genes, and it is controlled again from mutagenic region like H19 is right here, and then from inside the gene itself. And when you look at DNA methylation differences, when there is DNA methylation differences in either of those regions, what you see is a complete abrogation of these monoallelically expressed genes. Okay? And what we do know about some of this, like the DLK1, we know that they're associated with obesity, they've been associated with cancer. I mean, the relationship of actually DNA methylation changes there have been associated with liver disease as well. So what we did was we went back to the next cohort because we had seen that ecologically the highest incidence of liver cancer was in that region in Durham. This again is in Durham in Virginia, okay? And so the question then became, do we see higher DNA methylation in this region? This is based on about 600 people. That is your lead exposure that I showed you at the beginning. This is the cadmium exposure. What we're seeing here is hypermethylation measured, dichotomized at about the top quartile. And then take that top quartile, that's what was meant for hypermethylation. And what we're saying is that we see them coincide, but not all of the cadmium, but just some of them, right? And so the question was, why is that happening, right? So when we do the regression model, we are seeing the relationship, but it seems, in addition to that, that population that predominantly lived in that neighborhood I showed you is probably what seems to be driving the relationship. So maybe we are talking about a social stressor that we have not been able to measure. So these imprint control regions are really important. I mean, we discovered them a while back because of these diseases, right? In their worst form, when there is not just leakage, but when there is actual loss of imprinting, the diseases that come out of that are pretty devastating. Right? What we do know also is that when you see these imprinting disorders not as severe as what we saw with children, we do know that they are associated with liver cancer. So then the question is, are they overrepresented really? Are these imprint control regions overrepresented? And there has been some data showing that they're overrepresented in neurological conditions, in children and in adults, in metabolic diseases, and in some defects of function. But all this information here that I just said is just based on the 24 imprint control regions that we know of, right? So then the question is, where are the rest? Right? Where are these imprint control regions? If they're that important, we need to know where they are, right? They may actually help us characterize the exposome much earlier, especially in this periconceptional period that we actually don't know much of, right? So it also, is there any evidence that aberrant methylation, the difference from 50%, right, it associates with chemical and non-chemical? Do we know that? So the question is, where are they? Where are these regions? So we decided, you know what, I think we probably wouldn't be able to move forward in this DOHAD space until we were able to at least characterize them and be able to fill in that middle part. So what we then did was we decided, you know what, we're going to do this. We're going to take tissues that represent all the germ layers. We're going to look for the imprint control regions, right? Do some deeper sequencing and identify these imprint control regions. So to do that, these the approaches and fun process, but really what we were looking for was, we were looking for these differentially methylated regions throughout the genome that were around 50%. That were around 50% because you're expecting one allele to be 100 and then the other 0. So you're looking for 50%, but because there are lots of ways to get to 50%, right? You arrive at 50% by 25, 25, right? So the question is, how do you make sure that you are covering it? So then you go online, you go to the oocyte sequences, you go and look at sperm sequences, and then where you see those 50% marks, you are now looking for, do you see 0 or full methylation in those regions? So that's how we did it throughout the genome, and this is where we're now. So we actually do have, so this is an example of the next 3D one that is related to liver cancer that I showed you in that book with the. So this is what they look like. These are the methylation marks in those regions. You can see they are around 50% in all 3 germ layers. And then in sperm, you are seeing almost no methylation. And in the oocyte, you are seeing it, right? The oocyte data are very limited. Okay, they come from online. It's very hard to get. So this is what we used to know. This is the norm. I see the one that we have found. And this, remember, there were 2 of them. the methylation marks. And what we were able to find was this entire region actually is an ICU. So that is our candidate OPD device. So this is known, and then you go to all the places that were previously unknown and kind of look for similar patterns like that. And this is a region that is an example of the region that we didn't know before. So this is the place where you can actually go, the humanICR.org, to produce anywhere in the genome because it's embedded in the genome browser. You can go anywhere and put in your address and be able to find any gene you're actually looking for, any region you're looking for. And this was put together again by P30, and they're the ones that are managing the. So our center is actually managing this website. And this is an example of what can be done. So now we have put it on a platform, on an Illumina platform, or Illumina has done that. They approached us and they really wanted to do this. So we now have it on a platform. We take it, we measure in placenta because we already had measured environmental exposures. So then we look for this 1400 putative ICR, and we find that 31 of them, 82 are associated with Gutenberg specifically, among those that are also associated with arsenic blood concentration. And then among the 31 of those, they are actually shared. So what students are doing now is taking, and their basic science mentors, is to take 1 or 2 or 3 of these at a time and start interrogating them at the bench, specifically to try and figure out, are they really imprinted? Do they meet the other criteria for imprinting that you can't identify bioinformatically? In other words, do you see monoallelic expression here? Right? And this is another example of, this one is for liver disease. So these are 2 separates. So we have whole genome by self-and sequence, 51 controls and 10 HCC in blood. And this is what we have in tissues. And the question we were trying to answer here is, how much of this can help? How much of this is available? We know that there is differential methylation in tissue, right? That's what happens when disease happens. The question is, how much can we pick up in circulation that we can then see if we can use as biomarkers of disease much earlier than we are now. So doing this analysis got us to 1500 blood-based differential methylation. And this one got us to 600,000. And then overlapping these gets us to about 360. And this one overlap imprint control. It's a lot to look at, but we have a student that isn't to look at some of those regions, again, to determine whether this is correct. I mean, do you actually see them before liver disease happens, and do you see them when liver disease happens? And do they still look the same? And this I provided just so you see what it looks like on the genome browser. And these are the part of the ICRs that we put together, right? This is in the genome browser. These are the HCC related blood-based, and this is what you're seeing in tissue. So you're beginning to see that these do overlap in a way that you could actually use this as screeners with a little further development. And this is another example for a different disease. This is another example. Another example, a neurologist approached us because of this problem that all of us know with Alzheimer's. We don't have access to the brain, right? Just like we have. So we have clinical diagnosis that is pretty not good, right? And clinical symptoms appear much later than a lot of the processes that cause disease happen. So the question is, can you come up with, can this help us identify disease way before it happens. So the question then was, where do we get the tissue? So he provided us with autopsy DNA. As you can see, sample sizes are small, but it looked like African Americans are different enough from the white population that we wanted to make sure they were split. So we identified a lot more. So we started like, where are the differentiated regions? And then run those through your ICR panel to identify. And then these are the 120 that you end up with. And then you look at this and you say, who are these? There is very little overlap. Is this real? If you actually do an IPA to see what pathways are affected, it's the same pathways, right? But there are different regions that see them. At least the nearest genes, they're different, except for those two, right? And the two are one imprinted gene that we already knew. We know about it. This is new. The NLRP1 we didn't know about that before, but it's involved in the inflammatory. It's an inflammatory gene. And this again is an example of, this is what we find. This is what the ICR looks like. This is the known ICR, right? And then this is what we find in bones, and then so this is kind of, it's fun actually doing this, but question is, what does it really mean until we get to the bench and demonstrate that, yes, they are genomically imprinted, and yes, they are stable over time, and that they actually contribute to disease, we won't be able. But this is the very first step that our basic science community has been waiting for in order to select what regions to go for. So what we are doing now is refining these ICRs. As you saw, the way that we created them was the informatics person went and said, okay, this is 50%, and I'm going to chop it at 35 and 6, right? 35% and 75, 65, right? So you're saying plus or minus 10%, plus or minus 15%. That's not how it works, right? So what he's doing now is refining that a little bit and using change point modeling to actually say, is this region significantly differentiating affiliated compared to the next one, going through that throughout the genome. And then we have already using that next cohort, we are putting an arrow one that just got funded to look at these marks in children. And the reason we decided to do that is because the children had fatty liver, 10% of them had fatty liver at age 7 to 12. So what we are trying to figure out is whether we could have picked up some of those and maybe change diets in order to reduce the incidence. And a colleague is also working with the neurologist at Duke to try and find those that are related to Alzheimer's disease based on the data that I just showed you. There is a social epidemiologist at UNC who's also working on trying to figure out which marks are actually represented by that blob that I was saying. It shows that it's cadmium, but also being black seems like it's important. The question is, what is it about being black? So she's working on that. And also trying to collaborate with people at Adelaide who are trying to use these ICRs to try and figure out whether there is a way in which we can improve assisted reproduction. But it seems like they are archives of early exposure that we probably at a minimum use them in case control investigation. And that they can contribute to quantifying the proportion of disease that could be related to this early life period. Because right now we do know that this early life period is important. we see risk ratios that are statistically significant, but really what we do not have and I think it will depend on the disease. There are some that probably a very large attributable proportion is going to be realized related to these early exposures. But I think there will be some for which maybe they are not that important, that yes, they are significantly associated statistically, but really when you consider the entire, all the developmental windows out there, it may not be, some windows may not be as important. The other thing that the basic science we like about this is that they control multiple genes, right? As you saw with the DLK1, MEG3, they control up to like 14, 15, all the way from DLK1 to DIO3. This is like 1.5, it's a huge million bases. It's enormous. So all this, if you really think about it that way, then all you need is information, gene expression profiles that differ in, say, 50, right? And then you can pick up information about the rest. And I would like to stop here and wait for questions. Yes.
Take questions.
I mean, the reason I really wanted this to be more of a discussion, but then I started getting yes.
My question is, have you considered DNA sequence these genes? Because here is an example.
Yes.
Especially
the main actually. The way that we looked at them, one was to think about them as effect modifiers, right? And then in other ways, when we found a region that had too many genetic variants, we just removed one because it would have, as you said, it could be entirely, right? It could be, 50% methylation, but it could be entirely different. So this one here are specific. Yes.
I think on the definition of imprint control region, you did whole human sequencing, look at all three cell layers.
Yeah.
For 50% free.
Yeah.
I was wondering, could you have layers specific rather in one of those layers rather than not the other two, still being biologically significant? Or did you want to do all three just so you could look at blood and have a surrogate for other cells?
It is actually the latter. And also, if you think about if this matter laid down that early, they should be in all tissues so that we can look at blood, we can look at saliva, we can look at these inaccessible tissues and be able to use them as screening safe disease. And the choice of which, we know we had to look at all the regions, but the choice of tissue depended on what was available in image. They do have the tissues, and sometimes they have 5, sometimes they have 2. So the question was, in what individuals did they have them in tissues that they have?
Another question, like the Randy Journal website, the
Yeah.
That's like 2 or 3. Where's the other one?
That so Randy Journal's website looks for the actual intended, right? So if you look at that DLK1 MEG3 region, it controls, I think they are 15 to 20, right? So when you are looking at that website, you are thinking about the genes they control. They are controlled by an ICR. We have no idea where it is. So what we are doing here is trying to say, who is the culprit? Because that's what we can change. Yeah.
So I guess I'm just curious, how many ICRs are on the right and how many of them have been validated or known to be truly?
There are, this 1488, Illumina successfully designed 1100 of the ICRs. So they are still working on, because some of these regions are really difficult to do. So they are working on the remainder. And your second question is how many have been validated of those that we do not know, right? The ones that we know, I think many of them have been, not all of them, but many of them seem like they have been validated. Right now, this paper came out 2 years ago. There has been geneticists have validated, I think, because this is going to be a long slog, right? Because they're doing that, they are looking for monoallelic in this tissue, in that tissue, in that tissue. Okay, now we have done that. How about other criteria for genomic imprinting, right? So it's probably going to be a long slog, but I think population scientists can help by, and this is why Illumina is so excited to put it on the chip, because then you can run through a lot of samples and be able to just go for those that seem like they're checking out. You start with those. And that's why I think they were excited having putting it on a platform. In the charge a lot less than they do for their 850 or whatever that array is. This one here, I think we're charging less than that. So it makes it possible to actually look at that early period unencumbered by, less encumbered by costs.
And so I remember there's some slides you showed the geographical clustering, and that's associated.
Yeah.
And African has different ways population kind of back. So I was wondering, like, what is so is that like by environments such as those African races population maybe geographically clustering and different environmental exposures, or is that genetics, like the local ancestry different, and the fact that they change in ways that just longer.
I mean, the reason the Chantal Martin, the social epidemiologist, has taken this on is precisely for the reason to see, because it's hard for me to believe that it is that, because these metals don't conjugate, they don't. So I don't know how it would be a genetic problem. So I think what is happening is we have people who have multiple things. And maybe one stressor is important, but so in some could be just specific in some. Actually, one of the PhD students that is working with us, it was looking at something similar, where she was looking at the association between cadmium exposure and these results, right? Like placental size, preterm birth, and looking at it specific in African Americans, in Hispanics, in what she is finding. Overall, actually, if you have a small enough geographic area, there are no relationships. But you stratify that with the social stressor of and she's looking at adverse childhood experiences in financial stress. Now you start seeing that effect modification, yeah. But I think that because it's this structural compounding I was talking about, that you can't really remove these compounders via multivariate. You just have to be thoughtful about when you collaborate with others, what populations you bring into answer which stuff.
Technical questions that what's the third equation that
The what?
in there?
It may, but I mean, if you really think about it, when you start the analysis, you do not want whole genome based alpha sequencing data for one person, right? Because of the problem state. So you want to cluster them into 1, 2, 3, right? So that if you have somebody missing here, they're not going to be all missing here unless there is a technical problem, right? So if you were to say cluster 2 together, you pull them, you pull the DNA, you actually mitigate that problem quite a bit. The problem is that then you have a smaller sample set, because now you're pulled. But it seems to me from this bioinformatics people that it seems like it's a prepared strategy.
Fascinating stuff. Thank you so much. I'm wondering, in my world, I look at sources of exposure.
Yes. Yes. Yeah. Yeah. It's, yeah.
I just wonder if you sort of that in where are these exposures coming from? Because we have things like distinct files over their region.
I know exactly what you mean. I was actually really thinking of doing that with lead because that is so well, those isotopes are so well characterized. We tried to do it with that. We need a structural chemist. We can identify one.
I hear you.
But he has to get his tenure first, right? That comes from so you probably won't be available for a few years, but I very much agree with you. Yeah, that's the next question, really. There is structural compounding of the population, but there's also structural compounding of the chemicals themselves. Yeah.
So we have a question from online. They say, what are the next steps in your view of identifying true epigenetic responses to chemicals and identifying pharmacological?
I've thought a lot about that question. So I think people at the back didn't hear you.
Yeah, the question is, what are the next steps in identifying true epigenetic responses to chemicals and identifying pharmacological that are associated?
I think the first step is to appreciate that we're going to have different developmental windows and that they're going to affect us different. I think the first step is for, and this is not going to be me, but it's going to be other people kind of like thinking about what developmental window really makes sense. I mean, it doesn't have to be like each year or every 6-month period throughout the life course, but I think that we have seen from, it will be guided by epidemiology, right? Like what exposures occurring during what developmental windows tend to contribute to what diseases, right? And then I would then use that as a guide and then kind of like back into those exposure association disease relationships. And then back into that using these, whether you're using whole genome bisulfite sequencing or you're using these ICRs, if you're talking about very early in life. But it's not, I don't think that you can just go out there and look. I think you will look forever. I think you need to first define the window, and then you want to back into one of those windows with specific disease outcome relationships. Yeah.
You described what sounded like a frustrating experience with the mayor. Have you gone back to the mayor?
Yes.
Yeah.
Many times.
Yeah.
But the mayor has changed.
Maybe that's good.
Yeah, no, the mayor has changed 2 times since then, but it was also around the time of gentrification of the city of Durham, which and that's the reason why we thought this is a really good time, you know. But yes, it didn't go over well, but we are now engaged with the next mayor, and we actually got a lot of help from a Duke investigator who went and measured lead in the playgrounds. And that's always a lightning rod, right? And he found tons of it because, as you saw, SCEHSC should have found it first in that same community, and we found it, and this guy has found it. And that, I think it's in the last year, has resulted in some action. So I think what they're doing is mulching first and then blowing grass. Yes. Yeah. Actually, I've seen the studies in the adjacent county in Wake County. Not so much Chapel Hill. The problem with Chapel Hill is that the population is stable and it's aging. If you actually look at the demographic structure of Durham and Chapel Hill, it's like you're looking at Florida and someplace else, you know. And these are adjacent counties. I have no idea why, but it's not. I think Wake would be a good place to do the comparisons because both of them are dynamic. They have a lot of new people coming in. In fact, for the research triangle park and staffing that entire area and the 3-4 universities around that, all those people live in Durham and Wake. Nobody's moving into Chapel Hill anymore. So the population is a whole lot. Any other questions?
So I'm going to ask one more question about, it's interesting trying to take the translational research. This is kind of related to wrong. It's interesting trying to take the translational research and then apply it to make policy changes or drive policy changes.
Yeah.
Do you have advice on or thoughts on, it sounds like it didn't go well.
No, it didn't go well, but I think if you think about it, it's partly because of my naivety, right? I'm going to get a community person, and I'm going to get a person from our engagement board, and I'm going to, you know, then the mayor is going to organize a meeting. The mayor is going to be invited to a meeting, right? That's not how it works, and it shouldn't have worked that way. I think part of it was just being naive and so what has happened since is that our center has actually now developed community partners, not just one like we did before. We had the Children's Environmental Clinic, which was not run by Duke, but it was Duke related. Yes, it was situated in the community, but really it wasn't, they were not community partners. The person was the leader, but it wasn't considered community partners. What we have done since is to actually have relationships, not just with one, but multiple community partners. And that has helped not just the relationship, but when, for example, when the state engineer found that PFAS in the Cape Fear River, it was much easier. Things were a whole lot smoother and things went really well. So I think you can improve on these systems. And one thing I would say is never talk about animal models with the community.
Would you even talk about epigenetics with the community? I mean, would you try?
They actually understand epigenetics. They understand the epigenetics of trauma, right? They really seem to get that. And so you are replacing the trauma that they understand with another exposure that probably they hadn't heard of. But they understand that much better, that there are some marks that are established early, and some of them you even fail to erase, that they are transgenerationally inherited. They understand. That to me was an easier sell than the explosions. Yeah. Yeah. Any other questions?
Well, thank you so much.
Absolutely.
A really nice talk for people who are interested. If it's not too cold, there'll be food out in the courtyard.
Yes, and I'm really happy to collaborate. Thank you.